very excited about sharing tonight's program featuring Dr. Jason Hoeksema. But first, I would like to take this opportunity to remind everyone that the Conservancy is hosting our Drink a Beer, Save a River event on August 29th with Dale Sanders. If you're interested in joining us, please visit our website, wolfriver.org, for RSVP and registration information. I would also like to acknowledge all of our benefactors for 2025. They are the Crawford Howard Family Foundation, TC Energy, Bank of America, AutoZone, FedEx, the Hyde Family Foundation, Buckman, International Paper, the Assisi Foundation, Jim Karras Subaru, Terminex, Ring Container Technologies, Brother International Corporation, First Horizon Foundation, and Silvamo. Thank you for their ongoing support. Of course, all of our supporters, corporations, individual donors, and volunteers are critically important in allowing us to deliver on our mission, the protection and enhancement of the Wolf River watershed as a sustainable natural resource. As always, gifts of any size are appreciated. Please see our website at www.wolfriver.org/give. And finally, a few housekeeping details. We ask that you do not attempt to record this program with any device. If you have any questions during the program, please use the Q&A feature and not the chat box. Our Education Director, Kathy, will be monitoring the questions and will ask them after the program. Now, it is my pleasure to present to you our speaker for this evening. Dr. Hoeksema's research centers on plant-fungi interactions, and he teaches mycology at Ole Miss, Hotty Toddy, a popular course with students. He also enjoys sharing what he knows about shrooms with the public. Dr. Hoeksema earned a Bachelor of Science degree in biology from the University of Michigan, go Blue, and a PhD in ecology from the University of California, Davis. He held postdoctoral research positions at the University of California, Santa Cruz, and at the National Evolutionary Synthesis Center at Duke University before joining the faculty at the University of Mississippi in 2007. Ladies and gentlemen, Dr. Jason Hoeksema.
Eric, thank you very much for that introduction and for the opportunity to speak this evening. I'm going to go ahead and share my screen. Okay, so as Eric mentioned, I'm going to talk to you tonight about mushrooms. Specifically, the topic for this evening is finding and identifying wild mushrooms. And first, a couple of ecology lessons about the basics of fungi and how they interact with their environment. And one thing that a lot of people don't know is that although mushrooms are obvious, they're the macroscopic part of a fungus, I don't want you to forget about the other big part, which is the mycelium. This is the body of the fungus. And actually, it's the bigger part for most types of mushrooms. Mushrooms, in fact, are the fruit, the sexual reproductive structures of fungi. The mycelium is what lives inside the food that they're eating and is the persistent, usually long-term, long-living part of the fungus. And I've shown a photo here of a mycelium in the soil underneath pine trees on the coast of Oregon. You can find something like this by just scratching through the leaf litter in any forest system and in many other types of plant communities. Mushrooms, as I mentioned, are the fruit of a fungus or the reproductive structures. Here you can see a down log in a forest covered with mushrooms. Well, from the previous slide, remember that lesson, the interior of this log would be full of mycelium, the body of the fungus. But if you pick some of these mushrooms, that doesn't make the fungus go away. Even if you picked all of these mushrooms, that mycelium would still be there inside the log and likely capable of making more mushrooms in the future. And then finally, don't forget about the other microscopic part of a fungus, and that is the spores. So spores are like microscopic seeds, and they are produced by the mushroom, and they are, in fact, single cells that are small enough to be so small that we can't see them with the naked eye. When there's a single spore, if it's floating through the air, you can't see it. Some spores are big enough to see with the naked eye, but it's very, very rare. Most spores are microscopic. But you can see them when they're aggregated in a pile or a print, like I've shown on these images here. So this photo from my friend Pat Miller on the right is a pile of green spores or a spore print from a mushroom called chlorophyllum molybdites, where the green spored parasol mushroom, which is very commonly growing in our front yards, in grassy meadows, grassy fields. On the left, here is an orange mushroom that has left its own spore print out in the wild on the leaves that it was growing over. Can you see the white print at the sort of top left of that photo? And then in the middle is another example. This is from my front yard. You can see the blackish spore print on the cap of this mushroom left by the cap of another as it was overlapping it. And spore color is going to be important for mushroom identification. The best way to see spore color is to make a spore print or to watch for natural spore prints like you can see in those two photos on the left. The second major thing I want to emphasize in understanding fungal ecology is that it's important to think about what fungi are eating. What do fungi eat? And especially, where do they get their organic carbon and their nutrients? These are the two things that all organisms need to build their bodies. Some organisms, like plants and algae and some bacteria, can make organic carbon molecules from air, from carbon dioxide through photosynthesis, but fungi cannot do that. They have three main strategies for getting organic carbon, not photosynthesis. One of them is to be a decomposer, eating dead organic matter. And this example shown in the photo here is one such fungus. This is a decomposer fungus that eats wood. It's eating that log, and that's where it's getting its organic carbon and its other nutrients as well, like nitrogen, phosphorus, sulfur, and potassium, and so on. A second strategy for fungi to obtain their food, their organic carbon and their nutrients, is to be a pathogen or a parasite. This is a strategy where the fungus is eating other living things like plants. For example, the peaches on the left, the fruit on this corn plant here. So that's a fungus called corn smut, which is actually an edible fungus that some people really enjoy eating. It has this deep, sort of earthy corn taste. Some fungi can even eat other fungi. And then the third main strategy for fungi to get food is to be a mutualist, and that is to trade nutrients for sugars, for their organic carbon molecules. So I've shown you the two major examples here. Perhaps more familiar to you are the lichens. So on the right, those are good examples of three different species of lichens. Lichens are actually a partnership between a fungus and algae that is living inside the fungus and then usually growing on some sort of substrate like a rock or a piece of bark. On the other hand, mycorrhizae, shown on the left, are partnerships between fungi and plants. So what's shown there is the root of a plant like a pine tree, and all the little whitish tips on that root are mycorrhizal symbiosis points. That's where the fungus has colonized the roots of the plant. And then from there, the fungus can grow out into the soil and absorb nutrients and trade some of those nutrients to the plant in return for sugars. And that's how both of these symbioses work. In lichens, the photosynthetic partner is not a plant, but is an algae that's microscopic, but it's doing the same thing. It's photosynthesizing and giving sugars to the surrounding fungus. And in return, the fungus is absorbing things like phosphorus and nitrogen out of the rock or the bark that it's living on and giving some of those to its algae in return. And I emphasize these three strategies because knowledge of fungal ecology the mycelium, the spores, the mushrooms, and the ways that fungi get their food can actually help you find edible mushrooms successfully. So to show you some examples, uh, on the top left is an illustration of an oak forest where certain kinds of fungi like chanterelles are growing. In the middle is a painting of a field, a grassy meadow with puffballs. And on the right is a riparian bottomland habitat with morels. And these are three of the most popular uh, species of edible mushrooms, and each of them grows in a very distinctive habitat. And by understanding that chanterelles are mycorrhizal and only associated with trees, you wouldn't waste time looking for chanterelles out in a grassy meadow. And puffballs and morels have different habitat requirements, even though both of them are decomposers. Puffballs like to decompose the organic waste that's out in a meadow or a field, whereas morels are decomposing more woody organic matter found at the base of trees in a forest, in a bottomland, in a wet habitat. Um, Secondly, I want to talk briefly about strategies for collection of fungi, so that when you go out and look for mushrooms, perhaps tomorrow or this coming weekend after hearing this talk, these are some practices that you can put in place. One of those is to observe as much as you can about mushrooms in the field where you find them. And I really recommend taking photos of the mushrooms before you collect them. Uh, a lot of our phones now keep GPS points for where photos are taken. That's a really good thing because the location where a mushroom is collected can be really valuable information. Secondly, observe the habitat where the mushroom is growing. Observe the substrate, whether it's soil, buried wood, the trunk of a living tree, whether the tree is a conifer or a hardwood. Maybe it's growing on dead wood, the location geographically, and also notice what are the nearby tree species that are growing above the fungus or nearby. Third point is to collect whole mushroom specimens of multiple ages. One thing I'll try to show you some photos of in a little bit is that mushrooms can look pretty different when they're very young versus when they're old. And if you find several mushrooms growing next to each other, recognize that those are often young and old individuals of the same species. Collect all of them so that you can bring them home because the keys, the books that you use, will often refer to characteristics of young mushrooms or old mushrooms. And if you've only got one or the other, you're going to be a little bit in a tough place. Also, when you collect a mushroom, make sure not to just yank it out of the ground or break it off, but rather dig underneath with a knife and get the whole thing. Some mushrooms have roots with important structures on them that we need to have at home when we're trying to identify the mushroom with a book. I recommend storing mushrooms in wax paper. Ideally, wax paper is nice because it's a nice balance between it traps a bit of moisture, but not so much that it makes the mushroom sweat or get slimy. Paper bags are fine too, especially if you're just quickly collecting and bringing them home to eat. And then finally, observe as many characteristics of the fresh mushroom as you can, ideally taking notes so that when the mushroom changes, which they often do after collection, you will have good knowledge of what they look like before it changed, after you've bruised them, they've dried out, etc. And then here are some tools that are recommended. A basket is really the best tool for collecting mushrooms in because it's rigid and doesn't collapse when you have a heavy load of mushrooms, if you're so lucky. But canvas bag can be nice as well, or even just a paper bag. I usually keep a couple of paper bags in my car just for impromptu mushroom foraging. Wax paper bags can be bought on Amazon or elsewhere in the lunch bag section. Those are really nice. But if you can't find wax paper lunch bags or sandwich bags, you can buy just rolls of wax paper to roll up your mushrooms in and twist them to trap them in there, and then some brown bags. You may want a pencil and notebook. A sturdy knife is a must for trimming mushrooms in the field, for digging them up out of the ground, for cutting them off of logs, etc. Um, a digital camera, which is usually included now on any portable phone that you're bringing with you. A compass and whistle is a good idea if you're foraging in an area where you might get lost. That can help people find you. It can also be a safety device. A hand lens or magnifying glass can be very valuable, especially at home, but you might as well bring it with you in the field because it's small. And then watch out for unexpected problems in the field like rain, ticks, and chiggers. So bring bug spray. Consider permethrin treated clothing as a really nice option for keeping keeping ticks and chiggers off of you. If you only have bug spray, at least spray your shoes and socks and ankles and the cuffs of your pants. And then I recommend at least one good mushroom book. You really have to have at least one of these books if you're going to get at all serious about trying to identify mushrooms. So these are the three main ones I would start with. Mushrooms of the Gulf Coast states is maybe the most comprehensive for our area. Mushrooms of the Southeast is smaller, kind of a more pocket-sized book. It's a little thinner. It's easier to carry in the field, maybe in your mushroom basket. And those two books complement each other quite nicely. They don't cover exactly the same species. And then third, an oldie but goodie, is Mushrooms Demystified. This is a book, I think the last edition was from 1986, but you can still find it. And it's just full of gold on mushroom identification and collection techniques, and it has tons of information about different mushroom species. The names of mushrooms are getting outdated in some cases, but it's still really valuable. And if you read about the first 50 pages of each of these books, that'll really help you learn a lot about mushroom hunting and identification in general. And in addition, there are some great online resources. Mushroomexpert.com is perhaps the best. When you're trying to identify mushrooms, it has a lot of keys and good information about different taxonomic groups. Additional, more specialized books eventually might be nice to have on your shelf if you really get into this. And these are some recommendations. Mushrooms of the Southeastern United States is a really good book to have on the shelf to page through and look for species that you might not find in other books. Milk Mushrooms of North America is all about the genus Lactarius, which is super diverse and can be hard to identify. Similarly, there's this book on North American boletes. There's an additional Boletes of North America book that is also out now. And all of these books, these three and these three, I really recommend spending time just paging through them, getting to know what you're looking for by sight. Look at the names of these different things so that when you see something in the field, it might trigger some recognition and you can page back through the book and say, oh, I remember that one. Then you can find it much more quickly than using a key and starting completely from scratch. Also see some other online resources like amanitaceae.org if you're trying to identify amanitas. There's a Boletes of North America page on Facebook that's really active and useful. There's a website called the Bolete Filter that you can use to identify boletes, and so on. For our area, there's a nice active Facebook page called Mushrooms of Mississippi, which is useful up in Tennessee as well because a lot of the fungi are the same. And if you go on there and ask for help with an identification, you will usually get some good help from people. You can also just learn a lot by reading other people's posts. If you're going to post there yourself, I recommend giving as much information as possible, like the county and habitat where you found it, etc., the trees. Provide good informative photos. And by this, I mean close-up photos, clear photos that are not blurry. Don't send photos like I get from friends and relatives a lot of the time, where it's the person is standing up 5 feet away from the mushroom, and you can kind of barely see it, blurry in the distance. All we can see is the color. What we want is close-up, clear photos, not only of the top side of the mushroom, but also the underside, so that we can see the characteristics below. Perhaps cut through it and take a clear close-up photo of the cut side. Maybe get a spore print and show a photo of the spore print, a photo of the habitat, etc. Try to do your own homework on it before asking for help so that you can say, well, I thought it might be this, but please help me. And then as well, take any answers you get with a grain of salt. You know, there might be sometimes disagreement. You can't trust 100% of what you learn from people on the internet. And I think you probably all know that by now, but it is true with mushroom identification as well. I want to show you a couple of examples of good mushroom photos. Here's one. This is a mushroom I found growing in my front yard. Look at all that we have here. We can see the top side of this cap over here, showing it growing naturally, which is nice. Over on the right, I can see the underside of another individual. We can see the full length and color of the stem here. And then notice on this middle mushroom here, what do we see there? Well, that's a spore print from where this other mushroom was growing over it. So now we can see the spore color. The color of the mature gills, by the way, can often indicate the spore color approximately, but the best is to have a spore print. We get all of that, plus the habitat, which is the grass, all in one photo here. That's a really nice photo. Um, Here's another example. This is an Amanita species. One thing I like about this photo is it shows multiple ages. On the base, on the bottom left, is a very, very young individual that hasn't even expanded its cap yet. It's short and embryonic. Down here is a medium-sized cap showing the gills on the underside of the cap and the color of the stalk. And then we have mature individuals up here at the top in the center, showing the color of the cap as well as the color and the shape and the size of the stalk. This photo also shows the habitat, which is in a mixed hardwood conifer forest. And then here's a third example. This is a bolete. You can see the top side of the cap as well as the underside here, the stalk, and the bruising reaction where we pressed our finger into the pores and saw that it turned blue here. And we can see that this is a pine forest because of the surrounding habitat. So those are the kinds of informative photos that I encourage you to try to take. Now, a short lesson on mushroom anatomy. I've mentioned some of these words already, but let's review. So the top flat part of the mushroom is called a cap. Underneath a classic gilled mushroom are the gills. These are those thin, razor blade-like, flat, narrow structures called gills that you will find on the underside of a portobello mushroom or something that you will buy in the store. The stalk or stem is the part that connects the ground up to the cap. If there's a fungus growing out from the stalk down into the soil, that's called the mycelium. You may remember that from one of the first slides. Uh, and then we can have specialized structures, like up here on the stalk. If there's a ring around the stalk up here, that'll be called a ring or an annulus. That is the remnant from what's called a partial veil, which used to be covering the gills when this mushroom was young. And then some mushrooms have a remnant at the base, which looks like sort of a bag or sac. That's called a volva, and that's remnants from the universal veil that was covering the whole mushroom when it was young. That's something you can see on this photo a little bit here. There's a sac at the base. This mushroom has some scales on the cap, which are also left over from that universal veil. These mushrooms here also have a ring on the stalk left over from the partial veil. And when you flip over the cap of a mushroom on the underside, there are 4 or 5 options for what you might see, and you need to make note of this. Top left, we have gills. These are widely spaced gills compared to average, but that makes it kind of easy to see them. These are thin, raised, very delicate, floppy, sort of soft structures that break when you touch them quite easily. Underneath it, we have a chanterelle, which is showing gill-like structures, but these are not true gills. These are called false gills. Notice the difference in the structure of these. The false gills here are flat and sort of shallow and blunt and oppressed up against the cap. They don't stick out like a razor blade. They're not thin and floppy and easily broken. They're pretty tough and pressed up against the cap. Those are called false gills. Chanterelles only have false gills or no gills at all, like this individual on the far right. These are two different species of chanterelles. On the far right, we have the smooth chanterelle, which has no gills or false gills at all. It's completely smooth on the underside. Sometimes you'll find a slight hint of false gills on these smooth ones. Another alternative is in the middle here. This is a mushroom called a hedgehog, which shows spikes or teeth on the underside of the cap. This is a less common variant, but it is uh, it does exist, so you should know about it. And then on the bottom is quite a common variation, and that is for mushrooms to have pores or this sort of structure here, which looks like a bunch of holes. And this is characteristic of a couple of different groups of mushrooms, including this group here, the boletes, which have a cap and a stalk and pores on the underside of the cap. So I really encourage you to work to learn your vocabulary. This figure and this one are pages taken from David Aurora's book, the Mushrooms Demystified book that I showed you here on the right. It's got really nice illustrations, as do these other books. And what you need to do is spend a bit of time learning the vocabulary. Learn the different shapes of the cap that are possible, cylindrical, conical, bell-shaped, etc. Learn that there are different ways that gills attach to the stalk, whether not attaching at all like this, which is called free, adnext, sinuate with a curve there, adnate coming in straight, or decurrent coming in a down curve like that. There are different stalk shapes, different positions of the stalk. All of these things have vocabulary words that you need to learn if you really want to get comfortable with this stuff. Pour it over, and then when you go through the book and it mentions these things, you won't have to scratch your head and not remember what any of it means. And then you really need to learn how to use a key. All of these books that I showed you, most of them anyway, contain keys that work kind of like this. And you need to know how a key works. Most keys, a lot of keys, I would say, are dichotomous, like this one here, where you have to go through and make a series of choices from one or the other option. You start at the beginning and you choose the first or second option. So look at the ones here, and we're going to practice this. Let's practice this with the organism on the bottom left, okay? Bottom left with the green in the background, there's a little animal here. Let's use the key to see if it works for identifying that animal. Okay, say it with me, the answers here. Say it out loud to yourself or to whoever you're hanging out with there. Is the object not yellow? Okay, it's not yellow. So now you go to 4. See how that works? You pick 4. Now we go down to the 4s. Now you have to choose between these two choices, object purple and spiny or not as above. What do you choose? Not as above, right? So go to 5. Now the choices for 5 are object moving of its own accord when poked with 4 stumps or projections on its underside. That seems to be a pretty good fit. So we conclude that is a rabbit. So that worked pretty well. All right, let's see if this works for these other animals. Let's try it with the one in the middle. Okay, object yellow or object not yellow? Well, it is kind of yellow. Okay, so we'll choose two. Object more or less cylindrical. Uh, yeah, it looks like it might be. If you stretch that thing out, it looks like it might be a long cylinder. So we'll go with a yes. Okay, more or less cylindrical. So go to three. Object peeling easily. So you would have to grab this and try to peel it, or object not peeling easily. To me, it doesn't look like this object would peel easily. So I'm going to conclude based on this key that this is a banana slug. Now, how many of you think that this is a banana slug? Um, if you've lived in Santa Cruz, California, like I have, you would know that it is not a banana slug. If you've spent time in southeastern Arizona, you might recognize this as a black-tailed rattlesnake. So what's wrong here? What went wrong? Think to yourself, what is wrong with what we just did? Well, I don't see black-tailed rattlesnake anywhere on this key, but so what went wrong? Well, that's part of the problem, right? Is that the key does not contain the species we're trying to identify. And that is a fairly common problem with any key that you might use. So you have to keep that in mind, that in fact, you need to be making sure that you're using a key that includes or likely includes the species you're trying to identify. If it doesn't, you're going to go wrong and come to the wrong conclusion. What are some other issues that came up just now in using the key? Well, it seemed like some of our choices were not 100% crystal clear, right? Whether this is yellow or not, it wasn't quite clear because it's black and yellow. Is it more or less cylindrical? Well, the head is really not cylindrical shaped. So there were some problems. Peeling, how do you try to peel a rattlesnake? Well, that's not a very practical thing. So sometimes the key might not be written in a very practical or useful way for the organism you're trying to identify either. I'll let you go through the practice, seeing if this javelina here on the bottom right can be easily identified with this key. Um, okay, now with all of that background, I want to give you a bit of an overview about how I approach mushroom identification. And the first step I want to emphasize is called recognition. Step one. This is the best way to get started identifying a mushroom, and that is to recognize something immediately, at least to be able to place it immediately into its family or genus without having to think about it very hard. And you should be able to get there. The more you do this, the more you will be able to learn recognition. You will be able to look at a mushroom like this chanterelle here, and just because it looks how it looks, you will know that's the smooth chanterelle. This one on the bottom center, because it has a cap with pores on the underside and it has a stalk, it's in the group of mushrooms called boletes. And you can skip ahead to the bolete key in the book and start there. This one here, which I'm holding in the top of my hand, you can see that it has gills and it's exuding latex from where it's been bruised. It's exuding this little white liquid. Well, that tells you that it's in the genus Lactarius. And you can learn to recognize that quite quickly. And then you can go right to the Lactarius key in your book or to your Lactarius book without having to start from completely from scratch. That kind of recognition becomes really valuable to save you time. Okay, let's say that you don't recognize it though. You have no idea. Well, then you need to start from scratch. And here's what you need to do. First of all, as I mentioned before, note and photograph its characteristic using fresh specimens. Refrigerate fresh specimens overnight if you can't get to them right away, if you want to try the next day. Keep them in the fridge, not out on your countertop. Next, also, if there are caps that you're going to use to make a spore print, which is a good idea, see step three, then those specimens really can't be used after that for identification very well because they've been sitting out overnight. Similarly, specimens that you put in the food dehydrator are not going to be very useful for looking at their fresh characteristics. Number three, get a spore print. If you have no idea what a certain mushroom is, you need to try to get a spore print to ascertain the color of the spores. This is not required, but it is a really useful step because one of the best characteristics for telling apart major groups of mushrooms is their spore color. You can do this with only part of the cap if you only have one specimen. If possible, try to use a mature specimen, but one that is not too mature. Um, and you basically do this by taking a piece of the cap or the whole cap, cut off most of the stem, and then place it above a white piece of paper, maybe a black piece of paper, maybe both, and cover it with a bowl to keep the wind from blowing all the spores away. And after several hours or overnight, you should have a decent spore print if you get lucky. Sometimes, though, mushrooms will not readily drop a spore print, and you just have to shrug and try to use other approaches. Some shortcuts. The color, as I mentioned, the color of the mature gills can tell you the color of the spores to an approximation. Or you can watch for where the spores have been dropped onto the ground or onto other mushrooms and use that as a shortcut as well for the spore color. And then finally, to try to get to species, if you don't recognize the species right away, which you usually won't, use a key that matches your level of recognition. So for example, if you have no idea, zero idea what this mushroom is, start on page 9 of Mushrooms of the Gulf Coast States, page 29 of Mushrooms of the Southeast, or page 52 of Mushrooms Demystified by David Aurora. On the other hand, if you recognize that you have a gilled mushroom or a bolete or a polypore, start on page 112 of Mushrooms of the Southeast or page 59 of Aurora. If you know the family or genus, then great. You can start with an appropriate specific key in one of your books that is for that family or for that genus. If you're still stuck, try a separate book or a website for the group you're looking at. So if you know it's a bolete, go to the Boletaceae key. Or if you know it's a milk mushroom, go to the Lactarius book. Go to a book or a website for Amanitas, etc. If you're still stuck, then don't hesitate to just page through your books. Sometimes that works better than anything. If you get stumped, just page through the photos in all your different mushroom books until you find something that matches. And that can really be revelatory sometimes. I have found that there are certain mushrooms I could not identify except or until I just paged through the photos. And if you're still stuck after trying all of that, post some good photos on Facebook and ask some other people for help. Okay. Now, a couple of notes on edible versus poisonous mushrooms. I know quite a few of you are probably watching tonight because you're interested in trying to identify and collect and eat edible mushrooms. Well, here are some notes regarding that. First of all, many mushrooms are poisonous. Poisonous mushrooms can make you sick or dead. Some poisonous mushrooms are deadly, not all of them. A lot of poisonous mushrooms will make you ill in a way that makes you vomit. Maybe it makes you feel very nauseous. Maybe it's worse than that. Maybe it leads to a certain organ damage, but it doesn't kill you. Maybe it makes you so sick you wish you were dead. But there's a variety of outcomes. Um, but here are some things about poisonous mushrooms. You can handle them. There's no danger in touching poisonous mushrooms. They don't hurt you. You can even put a sample in your mouth to see what it tastes like on the tip of your tongue. Just make sure to spit it out. Just don't swallow them. Okay, now here's an important one. This is maybe the biggest takeaway regarding poisonous mushrooms versus edible. The only safe way to eat wild mushrooms is to be 100% sure which species you have picked and to be 100% sure if that species is edible. How are you 100% sure which species you have picked? Well, you can get there by being very experienced, becoming an expert, taking multiple classes, dedicating part of your career to it, like I have, um, asking uh, world experts, or just by focusing on a couple of easy to identify species that you have tried before or someone else has verified for you. Just focus on a couple of the easy ones, um, and you can be sure about those species. You can also ask an expert and they can verify for you. How can you be 100% sure if it's edible? Well, if you know that you have the right species, then you can look up the edibility in a book and you can take advantage of the many decades and centuries of experience that other people have undergone getting it wrong. Now, I want to emphasize there are no shortcuts or rules of thumb about edibility. For example, you can't just use your intuition and look at a mushroom and say, well, it looks edible because it kind of looks like those store-bought mushrooms. I hear this a lot. Somebody will bring a mushroom to me and say, well, is this one edible? It sure looks edible. There's no such thing, you guys. There's no such thing as looking edible. Some of the best edible mushrooms look terrible and unappetizing. Some of the most poisonous mushrooms look perfectly beautiful and innocent. There's no rules. You can't look and see that it's bruised blue when you touched it and therefore it's edible or therefore it's poisonous. None of that stuff works. All you can do is identify the species and be sure whether that species is edible. By the way, even edible mushrooms usually need to be cooked in order to be safe. There are some exceptions to this, like fresh truffles, and a few others, but mostly you should cook them to be safe. There are some chemicals that can cause a little bit of an upset stomach if you don't cook them. And then be aware that, as with any food, some people have allergies to certain fungi, causing them to get ill just like they would with other foods. And so you need to start with small amounts anytime you're eating a new mushroom for the first time. Now, with our remaining time, I'm just going to run through some common edible or poisonous species that you might find around here and point out a few key things about them. On this slide, we have poisonous mushroom number one, that is Chlorophyllum molybdites. This is from a lawn in Oxford. This is the most common poisonous mushroom we encounter in our area. It makes green spores and it looks like kind of like a store-bought mushroom, whitish. It can often look even whiter than this. It's got a scaly cap, a stalk, and a ring around the stalk. Uh, Second poisonous mushroom. Oh, and this one commonly results in mushroom poisonings because kids and others will pick them from their front yard. Hmm. Uh, I'm going to take a quick intermission to close the door because my dog is barking and I don't want that to interrupt us. Just 1 second. Aha, okay, sorry about that. Um, Edible mushroom number two. This is a group of mushrooms in the genus Cortinarius. Cortinarius, uh, they are difficult to identify and can be very poisonous, causing kidney damage and worse. They have two key characteristics. First of all, their spores are orange to rusty brown colored, as you can see by the color of the mature gills on the one on the top right. They also have a cobwebby veil on the stalk, which is called a cortina, and that's what they're named after. You can really see the cobwebby veil here. It's a partial veil just covering the gills. And when this mushroom fully matures, it'll leave that cobwebby veil as a sort of a fibrous ring on the stalk that often will catch those rusty when they drop down. So you can see that the cobwebby veil is rusty colored here. This purple one here, you can see as this cap is pulling away from the stalk, that cobwebby veil is still remaining there, but it'll become a ring on the stalk pretty soon. Okay, now here's some more poisonous mushrooms. These are two species that will result in your death if you go untreated. Amanita phalloides is called the death cap mushroom. It has not yet been verified in Mississippi, but it is found on the West Coast and the East Coast and a few spots in the interior of North America. And it looks quite innocent. It's nice and white on the underside with sort of a greenish cap on the top. Uh, and it is very, very poisonous. It is in the genus Amanita, which is characterized by having a universal veil and a partial veil. So look on the underside of both of these caps where they are going to have a ring on the stalk from the partial veil. And at the base, both of these are going to have a sac or vulva of some kind, which is a remnant of that universal veil. Those are characteristics of the genus Amanita. But then to identify these to species, you're going to need to use other characteristics like the spore color, maybe the shape and size of the spores under a microscope, the cap being kind of greenish here or all white here, the exact placement and shape of the ring on the stalk, the exact attachment and tissue composition of the vulva at the base of the stalk. Amanita amariverosa is the destroying angel, which does occur in Mississippi and Tennessee and is just as deadly as phalloides. And just one bite or two bites of these will result in death eventually. So we've talked about poisonous mushrooms, the dangers of them. So why would you bother then trying to hunt for wild mushrooms to eat if there are these dangers involved? Well, in fact, I believe very strongly that wild mushrooms are beautiful, fascinating creatures, and some are quite delicious. So it is really worth it. It's worth your trouble. And so let's go through some examples. Um, oh, and to illustrate the diversity, I wanted to use another painting from my good friend Melanie Roy, showing the incredible diversity that you can enjoy with mushrooms, even if you don't want to eat them. So notice that they vary in their color, their shape, their size. You can enjoy them macroscopically. You can look at them under a microscope. It can be like a delightful treasure hunt, even if you don't want to eat anything. And mushroom hunting is fun. You can bring your dog or your dogs. It can be a tromp through the woods on a beautiful day, regardless of what you're finding. So I'll end with talking about a few common edible mushroom species, and one that is not just one species, in fact, but is a group that many people in our area are interested in are the chanterelles. Chanterelles are probably the most recognizable, easiest to find, easiest to identify, and abundant edible mushrooms in our area. And in fact, there are several species that fall into this group. On the top right is the smooth chanterelle, which I mentioned before, which can have some hints of false gills, but is often very smooth, usually very smooth. Here is one that with false gills, a different species. Others grow in a more delicate clump, like this species in the middle here. Here is the red chanterelle, which has false gills that almost look like little gills, but it's got a reddish color and is usually quite tiny. On the bottom right in this group is the black trumpet mushroom, which is a really nice edible mushroom. What do you notice is common to all of these? Well, all of them have kind of a trumpet shape or flared shape to them. The orange ones have orange flesh on the outside, but if you break them open, they have whitish flesh on the inside. Also notice that none of them have a really distinct break between the cap and the stalk. And that sort of trumpet shape goes with that. And remember what I said earlier, they don't have true gills. They're either completely smooth, like this fragrant chanterelle here, or the smooth one here, or they have false gills, but they don't have true gills like you'll see in the gilled edible mushrooms. Okay, so chanterelles are ones to watch for, and I really recommend especially watching for them in the month of June. That's when we get the most reliable fruitings of them in our area after, especially if we've gotten good rain in the month of May, which is pretty consistent year to year. Chanterelles can also occur though in the mid to late summer or even early fall if we get enough rain. But if we get long periods of hot, dry weather like we've had recently, it can really shut down the chanterelles and you might not see them again until next June. Chanterelles are good cooked with dairy, with eggs, with pasta. They have an aromatic quality, not unlike apricots. And then when you cook them, they can be also a little bit peppery. And they're very delicious, just sautéed with butter and maybe a little bit of spice. There's some more photos of harvests of smooth chanterelles from our area. Okay, now here's a quiz for you. Uh, one of these is the black trumpet, and one of them is a little bit of a look-alike called the devil's urn. Without looking at the text on the bottom, which is which here? Well, you should recognize the black trumpet on the right. Notice that it has more of a flared lip and kind of a grayish, fleshy, almost a stalk underneath, whereas the devil's urn on the left is more of a cup with no discernible stalk, and it's more of a brown shape, and it has a sort of a crispy edge rather than that flared soft edge that you see on the black trumpet mushroom. Okay, let's move on to edible wild mushroom number two, which is the chicken of the woods. Chicken of the woods is actually two species that are fairly common in our area. There's sulphureus and cincinnatus, the orange one and the pink one. These are both polypores that grow on wood, either on upright trees or on fallen trees. Both of them have this kind of clustered shape where they grow one over the other. And in these heavy clusters that can be 10, 20, or even 30 pounds. Also, their flesh is kind of soft rather than really tough and leathery, like you might find with a lot of wood inhabiting mushrooms. The one on the left is pink on the top and white underneath, whereas the one on the right has yellow pores on the underside. Both of these are in the group of mushrooms called polypores, which has a shape like a shelf, but has pores on the underside of the cap. But there's no stalk. If it is a stalked mushroom with pores on the underside of the cap, then it's a bolete. But if it just grows on wood, like a shelf like this, and has pores, then it's a polypore. And these are chicken of the woods, which are really nice edible mushrooms that can be treated like chicken when you're cooking them, and are quite delicious. There is one look-alike called Berkeley's polypore, which makes these big, flat, whitish lips. And it's similar to these, but a little bit paler and without those bright colors, and it's also tougher. It's not poisonous, but it's also not good to eat. So just watch out for that one. It can be tempting to try to turn this into a chicken of the woods. Number three are the puffballs. I really like eating puffballs, and the best one in our area is Calvatia cyathiformis, the giant purple spored puffball. This one can get quite big, as you can see in the specimen in my hand there. And when you find them fresh, they are like a marshmallow on the inside, nice and clean and white and soft to the touch, but not puffy and spory. That's when they're edible. If you let them go too long, they start to turn puffy or liquidy and dark on the inside, and that's when you don't want to eat them. If they're dark on the inside, don't touch it. But puffballs found fresh can be really yummy. I like to fry them, like slice them and bread them and with egg and breadcrumbs and fry them just how you might uh, like a fried green tomato. And maybe make some tomatoes to go with them and make a little sauce to put on both of them. There are some sort of lookalikes for puffballs, like Amanita, when it's very young, is embryonic like this. It sort of looks like a puffball. But when you slice through it, you'll see an embryonic mushroom inside there with a embryonic stalk and cap. So watch out for that. You can see it emerging here, the amanita emerging from its sac, its universal veil. And then over here we have a poisonous false puffball, scleroderma, which looks like a puffball on the outside, but really when you cut through it, you see it's got a firm, dark interior, not a nice, clean, soft, marshmallowy white interior. Number four that I'll mention is the morel. Morels are prized for their edibility. They're my favorite, in fact, to eat. They're delicious, sort of earthy flavor and aroma, but they're tough to find in our area. The further north you go, it's easier. So maybe it's easier around Memphis than it is in North Mississippi, but it's sure tough to find them. My friends who are turkey hunters, some of them know where to find them because they have a short season during turkey season, and that is in our area, late March and early April. To find morels, you have to get out in the woods in bottomlands, like along the Wolf River, for example, in that specific time of year, and watch for them growing under trees like elms and ashes and sweet gums, and sometimes in weird places like in mulch piles and under privet hedges. But they're delicious if you can find them. But to find them, you need to get out and walk a lot in the woods during that late March and early April period. And if you can find them, good for you. Remember your spot and go back there the next year because they might be there again. Um, Easier to find in our area is oyster mushrooms. We have two different species, one that's more common in the summer, a smaller one that's pulmonarius, and then in winter, in fact, often in December around Christmas, we often find Pleurotus austratus. These are grow in a shelf sort of shape on dead wood, but they have gills, nice soft gills. So they're like a gilled mushroom growing on wood rather than a tough, leathery polypore. They're gray above with whitish, soft gills underneath. They'll make a spore print that is sort of whitish to sort of lavender. And they're very nice edible mushrooms and pretty easy to identify. Um, and then lastly, just a few other common mushrooms, some of which are edible, some of which are not. As I mentioned, the boletes are this really cool, diverse group that have a stalk and a cap, and all of them have pores on the underside of their caps. Some of these are edible, some of them are not, and they're tough to identify to species. Once you learn to identify the Strobelomyces, old man of the woods, that one's pretty easy. It's the only one that's this sort of purple scaly color, but others are pretty tough. But they're beautiful and worth paying attention to because some of them are delicious. Some of them are just super cool to find, like this one on the top left that has these bright red pores with little glistening golden dots of liquid on the underside of the cap and a ruffly yellowish red stalk. Super beautiful mushroom, one of my favorites. Um, One cool thing about boletes is that they often stain different colors when you etch into them and or bruise them. So you can make jokes like this. They're not the only mushrooms that turn colors when you bruise them, but they are among the best ones for that. Russula and milk mushrooms are super common. This one here is maybe the most frequently encountered mushroom in the whole world. It's found in all kinds of different habitats, different times of year. It's white stalk, white gills, and red on the top. It's not a good edible mushroom. It's too bitter and spicy tasting to be edible. That's in the genus Russula. It's got nice, firm, chalky flesh, lots of gills, and no other adornments. Similar is Lactarius, except Lactarius also produces a milky substance that we call latex when you bruise it or nick it or cut it. Some of these are edible, some are not. You have to identify them with the key, and it can be pretty tricky to get to the right species. Here's a nice edible Lactarius. This is Lactarius indigo, one of my favorite beautiful mushrooms of the forest around here. It's the only Lactarius that's bright blue like this and has a nice milky blue latex. And it often grows with chanterelles. It's not as common as chanterelles, but it is often with them and worth watching out for. And it'll stain your chanterelles in the basket. So here's a nice collection of chanterelles with some milk mushroom or with some Lactarius indigo um, interspersed there. Lactarius paradoxus is commonly found in the fall under pines. It's got this sort of variegated color ranging from purple to pink to orange, all mixed together. And that is a nice edible mushroom. It can be a bit slimy, but if you can pick them when they're fresh, they can be really nice and firm and have a good cooking texture. Another favorite Lactarius for eating is the candy cap. If you can learn to recognize this one, it's really great because when you dry it, it has a very distinct aroma of maple syrup, and it will infuse that flavor and aroma into anything that you bake them in. So I like to dry them on a food dehydrator, crumble them up, and then make cookies or bread with them. And those cookies and bread get this beautiful maple syrup aroma and a little crunch from the dried mushrooms. Um, so candy caps on the left. Here's an edible but bland mushroom called the lackluster laccaria on the right, which has white spores and whitish gills and a more cartilaginous stalk and an orange color instead of this nice subtle brown color. But be careful with this one because there are other brown mushrooms. You don't want to get it mixed up. There are other brown mushrooms that you can mix up with that one. So that's for learning from an expert. I mentioned Amanita, which is a genus that you need to be able to recognize. Amanitas are characterized by having universal veils and partial veils. Some of them, some species, the universal veil leaves scales on the cap, like this one here, or on the bottom right, the Amanita muscaria, or a big patch of flesh on the cap, um, or it'll leave a big sac at the base. And you need to watch for all of those possibilities. Some amanitas, like this Jacksonii in the middle, are delicious edibles, or this cocora mushroom, delicious edible. Some are hallucinogenic, like this muscaria, and some are poisonous, like this one here, this brunescens. Again, Amanita jacksonii. This is a nice edible Amanita mushroom that is quite distinctive. So this is, I would say, an intermediate level identification. Once you get comfortable, it's a pretty easy one to identify based on its distinct color of the cap and the stalk and this big white fleshy vulva at the base. Okay, quick quiz for you. Try to answer these for yourself as we go through, and then I'll give you the answer. Is this a bolete or a guild mushroom? Okay, hopefully you all said bolete. Is this Cortinarius or Cantharellus? Cantharellus is the genus of chanterelles, by the way. Well, it's Cortinarius because it's got the cortina, which is this cobwebby veil that covers the gills when it's young. And then what do we have here? Amanita or Russula? Well, what do you look for? Notice on this young one that it's got scales on the cap. And Russula doesn't have that. Also, it's got a ring on the stalk, which Russula does not have. Amanita is the one that has both scales from a universal veil and a ring on the stalk. So that's in the genus Amanita. Is this a true puffball or a poison puffball? Okay, good. That's the poison puffball scleroderma, which has that thick outer skin with a dark, firm interior. How about this? Is this oyster mushroom or Berkeley's polypore? Hopefully you all said oyster mushroom. Boletus or Tylopolis? Ah, I didn't tell you about this one. Well, let me tell you, Boletus is usually red or yellow colored, whereas Tylopolis usually has a nice earthy earth tones or subtle purpled colors. So this is a Tylopolis. Tylopolis would be a nice edible if they weren't so bitter. They have this beautiful aroma, but an unfortunate bitter taste that prevents them from being good edibles usually. All right. Well, with that, I will stop. And if we have time, I'd be happy to answer any questions that you might have. Thanks for your attention.
Thank you, Jason. That was just as wonderful as always. Um, yeah, we had one question so far, and that is, what is the purpose of false gills? But I'll ask also, what's the purpose of gills on a
Oh,
mushroom?
good question. I should have mentioned that. Gills and false gills, those are the zones on the mushroom where the spores are produced. So mushrooms are there to produce spores. They're reproductive structures, and it's the sides of the gills where the spores develop. And then they get shot out of there in between the gills out into the air where they get picked up by air currents and wafted away.
Okay. And false gills have the same, so they both have the same purpose.
Yeah, basically. Yep.
Okay. Okay. And then a question occurred to me, since we are a habitat protecting organization, I guess, and also just it's striking to me that there are mushrooms that come up in manicured lawns sometimes. So I guess I'm wondering about mushroom diversity in natural habitats versus urban landscaped habitats.
Hmm. Well, mushroom diversity is highest in our region. It's highest in forested areas. Um, and that can be young forest or mature forest, but especially in hardwood forests. Hardwood forests, mature hardwood forests around here have the best mushroom diversity, the highest numbers of species. Hundreds and hundreds of species of mushrooms are possible to be found in our mature hardwood forests. Pines and other conifers around here, pines being our most common coniferous tree, have some of their own distinctive mushrooms as well, but are not as diverse. And then there are mushrooms that specifically grow in grasslands and pastures. So those are going to have their own unique species. But in manicured habitats like lawns, we will have fewer. There are some that grow in grassy lawns and in potted plants, for example, uh, or even in, mulch beds and around our landscaping, but many fewer. The more native trees that you use in your landscaping, um, and the more large trees you leave in your habitat and your property, the more mushroom diversity that you will also have.
Okay. All right. Another question. First she says, fabulous, thank you. And what do you mean exactly by a mushroom being fresh?
Fresh?
Mhm.
Uh, well, uh, so when you find a mushroom, it can be in any state ranging from just popping out of the ground to fully mature to past its prime or starting to decompose. What happens is that over the course of several weeks, well, it depends on the species. There are mushrooms that do this over the course of days or hours, and others over the course of weeks, or others in the course of years. They mature and then they rot. And so if you find one that is still maturing, or at its peak maturity, then it's fresh. And if it's past peak maturity and starting to get soft, starting to shrink, starting to dry out, starting to rot, then it's no longer fresh.
Okay. The next question is just wondering if a link will be available so we can view the entire talk. And yes, in about a week, we'll send you the link to this recorded webinar. Okay, so next mushroom related question, where are spores on smooth chanterelles? Good question.
Oh, yeah. Well, it turns out that they are born on that smooth surface. Those mushrooms have evolved a strategy of just making their spores unprotected by gills. And it's you can speculate about why that might be. In gilled mushrooms, we think that gills are there to protect the spores from getting eaten by things like slugs and insects like little collembolans. But chanterelles are just not bothering with that and making their spores right on that underside of the cap, not on the top, but on that smooth underside.
Pretty cool. Do we have mushrooms in the South that you see only after fires?
Yeah, there are some, but two things. One is that I don't know a lot about them personally from my own experience, partly because I have not spent a lot of time mushroom hunting after fires around here. Secondly, there are more fire-specific mushrooms out in the West, in the Western United States. There are lots of really fire following specific fungi in the Western mountains, in the Rockies, and the Cascades, and the Sierra than there are here. Um, but it does occur here. It does happen.
Okay. Next question. I loved your presentation. Thank you. As a hobbyist mushroom grower, which mushrooms in your presentation can be grown indoors?
Good question. Well, as a rule of thumb, mushrooms that are mycorrhizal, relying on trees for their growth in nature, cannot be easily grown anywhere else. So these boletes, all boletes are mycorrhizal, cannot be grown. Oyster mushrooms, on the other hand, can be grown. Here, let me come back to the edible, to some of these other ones. So, uh, candy caps, on all other lactarius are dependent on trees. Can't grow them. Similarly, Russula, you can't grow them because they depend on trees. Boletes are all dependent on trees for the most part, and so you can't grow them. Even the best edible ones, you have to find them in nature. Oyster mushrooms you can grow. You can buy kits online to grow oyster mushrooms in your closet. Because they eat wood, you can feed them with wood chips or sawdust or the like. And you can buy a prepackaged kit to do that at home, and they can be cultivated. So if you find them in the grocery store, they're almost always have been grown in a facility somewhere, not harvested wild. Morels, I've heard of somebody having success growing morels, but it was very difficult and they don't taste that good. They don't taste as good as the wild ones. So if you find morels in a grocery store, they're almost certainly picked in the wild and you can't bother to grow them. I've bought a kit before, but the kit has to be installed in your property outside, and you'll use a bunch of things like sawdust and charcoal dust and blah blah blah, and then it won't work. At least that's my experience. Um, Amanitas, you can't grow them. Puffballs, I have not seen cultivated. In theory, it seems like you could grow puffballs, but I've never seen them cultivated. I've not seen them for sale in the grocery store. So that'd be interesting to try. See if you could figure that out. Chicken of the woods, I've never seen cultivated either, but in theory, it would be possible. They grow on wood, so maybe on sawdust or something, but I've not actually seen them cultivated. So, um, yeah, I don't know. And then chanterelles, nope, you can't grow them. They're all dependent on trees in the forest. So if you find chanterelles being sold, they've been picked in the woods by someone. This photo here on the left is a photo I took on the side of a highway in Poland when I was there a few years ago. Somebody had gone out and picked a whole bunch of chanterelles in the forest, and they're selling them by the basket there. And you can find chanterelles for sale around here at the farmers market, the Midtown or the city market in June. and sometimes other months, and sometimes on local menus at restaurants. They'll have them on the menu, but only seasonally usually. And you can find them in certain grocery stores at certain times of the year. I think Chicory Market had some this year, and in Oxford, and Whole Foods will have them sometimes seasonally, along with morels and some of these other edible ones.
Mhm. Okay, next question. Do jack-o'-lanterns grow in Mississippi and the Mid-South? They can look like chanterelles.
Yeah. Jack-o'-lantern mushrooms do grow here. They're not that common, but they do occur. Things to watch for there is that jack-o'-lantern mushrooms will have true gills. Um, Also, they're thicker and more fleshy. They don't have this sort of thin, wavy shape that chanterelles have. They don't have this trumpet shape. They don't have decurrent where this decurrent shape where the false gills run down. They'll have true gills and a true cap that looks distinct from the stalk. Usually, also jack-o'-lantern mushrooms are growing on dead wood, stumps, and the like, and morels or chanterelles don't do that. Chanterelles are always growing out of the ground, out of seemingly just out of the leaves and the soil.
Hmm. Okay, and this looks like maybe our last question, I'm not sure. About what percentage of mushrooms are edible versus unedible?
Oh, that's a tough question. Yeah, if I had to ballpark it, I would guess that something like maybe a third of mushroom species are poisonous, and a third are edible, and another third are sort of unknown. Um, uh, in terms of deadly ones, it's probably more like, less than 5% of species are deadly poisonous. Most poisonous species are not deadly poisonous. They're just going to make you throw up and feel crappy for a few hours, and then you just need to get rehydrated and you'll be okay.
Yeah. Did we, someone missed the beginning of the program and wonders if we discussed wood ear.
No, I didn't mention that one. Um, we do have them around here, and I didn't include it in the part of the presentation that I gave already. I may have seeing if I have a couple of slides here. I have a few additional slides, and I don't think it's occurring in those slides. One that a lot of people want to find around here is lion's mane, which grows on usually up above the ground pretty high. Looks like this nice white toothy one. And those are fairly easy to find in December if you walk through the woods enough. Um, yeah, wood ear mushrooms I don't have pictures of, but those do occur around here. They sort of look like gnarled, slimy, rubbery ears, and it's worth watching for them for sure.
Are they edible?
They are, yeah, yeah. They're delicious. They're kind of rubbery. If you've ever eaten seaweed, fresh seaweed, they have a very similar texture to those. But they're often used in Asian, various Asian cuisines. And they can be dried and rehydrated or used fresh. And they're good with rice and stir fries and that kind of thing.
Mm. So my last, this isn't a question, but just a comment that I'd like to come and have dinner at your house sometime. Have some mushroom cookies. It sounds great.
Well, you're invited anytime, Kathy.
I'm
One
just
thing
kidding.
one thing I'll just mention is that one thing we're pursuing is trying to learn to find pecan truffles. So if anyone has a pecan farm or access to a whole bunch of pecan trees, there's probably pecan truffles that grow there.
Oh,
And I'd love some time to take my dog, who is somewhat trained to find them, to look for pecan truffles in your orchard, and then we'll add a new wild mushroom to our palette around here. That's one that I've not found in the wild yet, but I know they occur. We've found the DNA on the roots of pecan trees in the Delta in Mississippi, and we're hoping to start being able to find them pretty soon as well.
well, that's great. And so I'm sure there's some pecan grows out there among our viewers. I hope so. Um, Jason, thank you. This was just outstanding as always. Just great information. And I think I'm going to turn this program back over to Eric to see us out. And I'll see you soon. Okay.
Okay, thanks again.
Well, thank you, Dr. Hoeksema. I appreciate that incredible presentation and your time this evening. And I'd like to remind our viewers once again, we are hosting a Drink a Beer, Save a River event on August 29th with Dale Sanders, and we'd love to have all of you out. Please visit our website at wolfriver.org and check our calendar link there for all of the details. Thanks again for joining us and have a wonderful evening.